Flip the switch. There we go. Now I can hear myself. Good morning, everybody. We'll probably have people trickle in based on the weather and the roads, but this is spring in Minnesota. I'm Aaron Kelly, and I'm going to be introducing our speaker this morning, and it's an honor to do so. Dr. Ellen Demerath. She is a professor in the Division of Epidemiology in our School of Public Health here at the University of Minnesota. She did her undergraduate training at Harvard, and then did her master's at the University of Pennsylvania and went on to do a PhD at the University of Pennsylvania. Did her postdoctoral training at Wright State University in the School of Medicine in Dayton, Ohio. Ellen has multiple R01s as PI and a very robust research program in areas extremely relevant to what many of us do here in the Department of Pediatrics. Her work focuses on the interplay of genetic and early life and environmental factors on growth rate and body composition changes in infancy, adolescence, and adulthood, and their impact on obesity and chronic disease risk later in life. Her studies cover multiple themes, but most notably developmental origins of cardiometabolic disease, genetic and epigenetic determinants of obesity, and methods for body composition assessment. She has well over 150 peer-reviewed articles and book chapters on the epidemiology of obesity and related risk factors. And notably, she serves as the co-director with our own Dr. Logan Spector for the Driven to Discover research facility at the Minnesota State Fair. So she's a lot more like us probably than those folks over in School of Public Health. Anyway, without further ado, Ellen, thank you for coming to talk with us this morning.
Hey, thank you so much, Aaron and Logan, for inviting me, and Dr. Neglia, Dr. Bender, all of you. Thanks for coming out at this time of the morning in spring in Minnesota. So yes, I am in the School of Public Health, and I guess I'm a little bit more like you because I do take a very biological, clinical sort of perspective at times, and yet also I try to weave in a population level perspective as well. And just for the disclosure, I have no financial or other disclosures to discuss. So my interest primarily is in obesity, so obesity prevention, childhood obesity. And as you've been watching, you know, Cynthia Ogden and her colleagues very frequently come out with these estimates of the prevalence of obesity in adults and in children. And for a while we've been saying, oh, you know, it's kind of coasting, it's kind of the same, but at least the young kids are doing better. The 2 to 5s, we had a little bit of a down, up and down, but kind of down trend, right? And people are like, well, that's because we in public health are doing our jobs. We're out there getting people to, you know, breastfeed and take the elevator, and then when they're pregnant, they won't gain as much weight, and so that's why we have this. Well, just last week in JAMA, we just got the bad news that the preschool obesity rate, 2 to 5-year-olds, back up to where it was in 2004. So this bings around a lot. Actually, this is data, so it's a little bit small sample size, honestly, even in enhanced. Nonetheless, we're not doing great. It's still kind of hanging tough, and obesity is very resistant at the population as well as at the individual level. So if we're thinking about prevention and early childhood, I mean, my mind always goes to the previous generation, right? What's happening in that family? 20% of women today are pre-diabetic, reproductive age. 5 to 10% pregnancies, depending on where you're looking, are affected by GDM, and a full 50% of women giving birth in the United States are either overweight or obese. Okay, so back in my mom's time, 19%. Okay, so it's skyrocketed, and certainly we know that that probably has a lot to do with what's going on with children today. Women who have gestational diabetes, their offspring have at least about 1.5 times greater risk of obesity in childhood. Women who are obese, their offspring have about a 2 to 3-fold higher risk. And a lot of the studies are seeing that this GDM relationship actually is mostly driven by the obesity that is predisposes to GDM. But then we get into, the biology. So why, right? I would say a lot of the work that in my area of, in the School of Public Health rather, not, but my colleagues, is focused a lot on sort of child behavior, diet, exercise, and what's happening in the home with the parents. This sort of very much postnatal behavioral approach, right, to obesity prevention and treatment. You sort of change your diet and increase your activity. But there's a lot of sort of prior factors that are involved in that transmission of obesity from parent to offspring. First of all, we're all living in the same environment as our children. So, for instance, pollutants, air pollution is an obesogenic factor that both parents and offspring would share, as well as their shared genomes. And then in the case of the mother, there's the intrauterine period, which has major programming influences on obesity, which I will not speak about today. It's a huge field that you all are very well aware of, and there's been a lot of work for at least 20 years in this. And more recently, we're learning about the microbiome, all those microbial communities that are in us, on us, that we now are recognizing are actually just not along for the ride, but are active players in our metabolism. But there's another factor, another sort of mechanistic link here between parents and children, or at least mothers, and that's breast milk. So milk, you could say, is sort of the next biological, true direct biological link after she gives birth to her infant. It's a direct link there between her biology and the infant's biology. And we know that breastfeeding women or obese women tend to have a higher rate of formula feeding. Okay, probably mostly aware of that. And although I would say the public health approach is to think about that as an issue maybe of education or social support, all of which is very important for the decision to breastfeed, we also know that there's a lot of physiological factors that are driving the for obese women and diabetic women as well, which I'll get to in a sec, that hinder breastfeeding. So maternal obesity is been found to have a delay in the prolactin levels, which, as you know, are very important for instigating lactation. They experience a low milk transfer after birth. High leptin, actually, we know that obesity, of course, is associated with high leptin levels, right? Leptin inhibits oxytocin. So that can hinder the milk ejection process. So there's each increase in BMI delays lactogenesis 2, which is the phase of lactogenesis when you're producing milk after parturition. And women with high BMI have much shorter breastfeeding duration. Some of these things are also true of gestational diabetics. There's that delay of lactogenesis 2, lower milk volume. We think that some of the hypoglycemia in women up and down on their glucose levels, that actually can reduce supply of energy to the lactocytes, inhibiting lactose and lactation. And so they're just overall less likely to breastfeed. And like I said, so we know there's some social and behavioral issues there. So some of it is lower body confidence. One of my graduate students wrote a nice paper about how obese women tend to have lower body confidence and feel that they aren't going to be able to breastfeed as successfully. There's issues of modesty, of being concerned about breastfeeding in public for a woman who is larger. There's also, though, physiological obstacles. And like I said, delayed lactogenesis 2 and lower prolactin, and also some very interesting insulin-related processes there. We think that the epithelial gene expression that sort of turns on the production of milk in the lactocytes is insulin dependent. The insulin receptors go charging on an expression on those lactocytes and in the mammary tissue cells during pregnancy, and especially right after birth when progesterone levels go down. And we also know that there's some interesting differences in the mammary gland development itself and its differentiation, going all the way back to puberty in those that are obese and non-obese. Most of this we know from animal studies. Really interesting area. Hasn't been as much done in humans, but very interesting. Okay, but let's step back. What about beyond just the quantity of milk, the ability to produce milk? What about breast milk quality or composition? Breast milk is incredibly complex. There's over 1,000 components, most of which we think are active, but we don't know their function yet entirely. So, of course, there's the gross composition. There's the stuff we measure, especially the fat, the protein, and the lactose, but especially the fat is variable. Fatty acids, all of you in the room who study neurodevelopment, very interested, right, in DHA and AHA and those other fatty acids that are important in cognitive brain development. But beyond that sort of nutritive side, there's all these non-nutritive things like hormones and especially immune factors, antimicrobials that are in milk. Those immune factors we know play a role, right? So breast milk is protective against all sorts of gastrointestinal infections. But some of these other things like hormones, the microbiome, we used to think that breast milk was sterile. It is not sterile. A whole microbiome, bacteria, virus, maybe even fungi that exist in milk. Environmental pollutants get in there. And there's if you're tracking the literature, I'm sure you're hearing all about human milk oligosaccharides, those, 3 to 10 chain saccharide molecules that are not nutritive for the infant, but they're supplying nutrition to the microbiome that's living in the gut of the infant. So all this stuff is in there, and we're sort of on this brink of understanding a lot more. However, what actually influences that is really not terribly well understood. There's all kinds of different types of factors. For instance, we know, at least from the gross composition standpoint, that breast milk fat fluctuates during the day, okay, higher in the morning. Lactational stage, transitional milk and colostrum, different from each other, different from mature milk. Stage of nursing. When you start nursing, you have a sweet milk. By the end, when you're getting the hind milk, much fattier. There could be differences in milk by gender. This has been shown in some animal studies. Birth weight, a larger child is going to demand more milk and perhaps change its composition. There's all sorts of behavioral factors, the time since the last feeding, and then many maternal factors that we just don't understand. The maternal diet, we know her fatty acid intake in the diet will be reflected in the milk's fatty acid profile. But a lot of these other factors we really don't know much about. Existing studies, mostly small, and I would say they're not very well controlled for some of these other issues that we know to be important. But why do we think this is important? Well, it's not just out of the blue that we think that maybe hormones and inflammatory factors, et cetera, could be part of that link between the mom's obesity and the child's risk. This comes from animal studies, right? So diet-induced obesity in rat dams leads to 300% higher leptin levels in the milk, 130% higher insulin in the milk, and so on. And then those offspring that are feeding on that obese dam's milk themselves become obese. Now, the nice thing about animal studies that we can't do with humans, although there have been some clever studies that kind of get at this, is the cross-fostering thing that we can do with animal studies. So to isolate the lactation part, what we do is we cross-foster the offspring of the obese dam onto a lean dam who nurses them, and the other way around. And so what you see is that the offspring of the obese dam that you'd think should be obese, if nursed on a lean dam, becomes completely normal, indistinguishable in some of these studies from the offspring of the lean dam themselves, who was nursed by the lean dam. And you can sort of fix the problem by taking that obese offspring and doing oral supplementation of leptin. Okay, that can reduce appetite and reduce body size in that offspring. So there's really good kind of mechanistic animal level research that indicates that this idea that hormones in milk could be bioactive for the offspring. That quasi-experimental study I was talking about in diabetic women, what they did is, this is a German study done about, 10 years ago, more than that. And they had all diabetic women, but then they offered non-diabetic donor milk to all the women that if and when they decided they wanted to supplement, to the amount that they decided they wanted to supplement. So it wasn't randomized, but it was sort of quasi-experimental because all the breastfeeding women, many of them, not all, many of them were supplementing with non-diabetic milk. And what they found was that those who were exclusively nursing with their own milk, their children had a 2.5-fold higher risk of overweight at age 2. And that there was a sort of graded response. So the more you supplemented with the donor milk, the lower your weight at age 2. Another thing just to point out is a little bit about the biology of milk synthesis. So here we are. We're in the mammary gland. We're in the mammary bolus here. And so this is all operating in these epithelial cells here, where the genes are turned on to start producing milk. But the mammary tissue needs to take its substrate from somewhere, and it gets it from the blood supply of the mother, as well as the surrounding tissue, right? So including adipocytes. The breast tissue is very fatty tissue, and the breast milk is actively produced. It's not just passively taking substrate from the surrounding tissue, but it's coming in, right? And so this is another reason we know that in obese women, we've got high leptin and high insulin. So that is coming, it can come right into the milk, all right? There's no reason why it can't. So what we decided, my colleague David Fields and I at the University of Oklahoma, is that it was time to do a kind of more formalized large-scale study of these phenomena in humans. And so aim one of our study, which is ongoing now, it's in its 4th year, is to look at the relationship of maternal weight status before, during pregnancy, and then also in the postpartum period to milk hormone and cytokine concentrations. The goal is 360 mother-infant dyads. The study, by the way, is called the MILK study. And what we're doing is we're following mothers and infants from their about second to third trimester of pregnancy to 6 months after. And we're doing serial milk sampling and infant body composition assessment. I just found out about a month ago that we're completely done with enrollment. We've hit our targets. So now we're just in sort of follow-up phase. Okay, at this point, I'm going to be talking about data coming from 220 of these women. So this is the number that we've got, all the assays run and the data on that I can analyze at this point. But I really want to point out that what you're going to be looking at here is the milk of healthy women. All right, so there's going to be obesity. We're specifically looking at obesity, but other than that, we try to get these women by inclusion and exclusion criteria to be healthy. Okay, so obviously all of them, they're in the Health Partners system, so they all have good prenatal care. They all have data on their BMIs going back to at least 8 weeks. So we know they've been in there for their prenatal visits. Okay, they're non-smoking, they're non-alcohol drinking. They're able to complete forms online. All right, so they're generally well-educated. That's not a restriction, but they turn out to generally be well-educated. And the most important thing, I think, from a design standpoint, is that for milk sampling, we require that they are all exclusively breastfeeding at their 1-month visit. So this will enable us to parse out that which is supplementing with formula, which may impact the milk, which is more frequent, right, in the obese woman, from the milk itself in the obese woman versus the normal weight woman. So all the women, regardless of their BMI status or their gestational weight gain, are exclusively breastfeeding. And I didn't just mention that we use skim milk. We do a very standardized milk collection. I can talk more about that if you're interested. Really rigorous control over the timing of the milk collection. And then we use your commercially available ELISA assays to measure the hormones in the milk. Okay, so the first thing is that we don't know so much about serial changes. We know, that the colostrum is really different in terms of protein and immune factors than the mature milk. But as far as these hormones, we don't know a lot. So our study is just starting to describe that. That these hormones tend to be higher at 1 month than at 8 months. Leptin, adiponectin, insulin doesn't change. CRP goes down. CRP is a C-reactive protein. It's a acute phase reactant that is stimulated during infection. It stimulates the production of all kinds of cytokines. IL-6 is one of those, and that decreases over time. So, take a drink here. We're going to go now into the phase where I'm going to talk to you a little bit about some of our findings on AIM-1. So, this is probably the one finding if you're thinking about what do we really know about hormones in milk and how they relate to obesity. This is the relationship that you find over and over again in the literature. This is not novel, what I'm showing right now. Leptin in milk in humans follows exactly what we saw in the animal models. You have a 2 to 3-fold higher, these are log levels, but a 2 to 3-fold higher level of leptin in the milk of obese women than normal weight women. So just animal models would predict. The two different colors are those with gestational weight gain within the guidelines and those that exceeded the guidelines. And so generally track the same, but in the normal weight women with normal weight at pre-pregnancy BMI, those with excessive gestational weight gain actually have significantly higher leptin. Okay. We also found a relationship with postpartum weight loss. So at this end is the lower postpartum weight loss. These women lost more weight after they gave birth. And we see, especially at 3 months, by 3 months where they've lost more of their weight, we see a decrease in leptin with greater postpartum weight loss. Insulin, again, similar, not as strong as leptin, and I think that's the reason why it hasn't been replicated as much, but there's been a couple studies now that show in humans, as in the rats, that there's a higher level of insulin in the milk of obese women. And milk CRP. Again, you're seeing sort of this similar situation, right, where you've got higher levels with higher BMI. And in this case as well, with CRP, we have an independent effect of excessive gestational weight gain, but mostly seen in the normal weights. We are also trying to get at other factors besides hormones, and we've begun that work. We're going to continue it hopefully in a renewal period, but we did a first pass on a subset of participants, 35, where we did LCMS to identify metabolites that would differ in normal weight. Those are the open circles, and overweight obese women. Those are the closed blue circles. These, this figure right here shows all the 9 different metabolites that differed by maternal weight status. Notably, 3 were those human milk oligosaccharides. Okay. And then there's other factors, amino acids. And there's a few that also are associated with infant weight gain. So this is very preliminary. It's actually in press with American Journal of Clinical Nutrition right now, but it's quite preliminary, and we hope to follow up to do a lot more work on metabolomics in milk. Okay, so that's so cool, right? So we're seeing some similar stuff as we see in animal models, and we're replicating things that have been shown in the literature, pushing it ahead a little bit. I also didn't show some adiponectin results that we also have some variation in. But does it really matter? Does it matter for the patient? I mean, there's plenty of stuff that varies in the human body, and it isn't all terribly relevant to disease or to outcomes, right? I gotta love this. This is one of our milk babies and her dad, who was willing to get photographed for stuff. So this little figure comes from a recent review on human milk composition and infant growth. So just like there's all these things that can differ depending on maternal metabolic status, all those same things may be important for infant growth, but we don't. We know something about HMOs and the microbiome. We know quite a bit more about macronutrients, micronutrients, vitamins. We don't know so much about hormones and cytokines. So those are the things we're going to talk about. Now I'm going to focus on leptin, adiponectin, insulin, CRP, IL-6, and outcomes being infant anthropometrics and fat-free mass and fat mass. And these data are newer to us, and so we're going to be focusing on 1-month outcomes primarily. Okay, now remember leptin, right? It's higher in obese people in the serum and in the milk, but remember what leptin is supposed to do. Leptin is supposed to suppress your appetite. It goes up after you eat, right? And it's that signal to the brain, up, we're done, satiety. You can stop eating now. So we hypothesized that milk leptin would have an appetite suppressing effect and perhaps therefore a body mass reducing effect. Okay. So we did find that, and these are sort of small effects, we did find a significant association, tertiles of milk leptin here. That in the highest tertile, there was lower fat mass in the infant at 1 month and lower percent body fat. We found also negative associations for milk CRP with infant fat-free mass, shown here, length for age, as well as weight for age. Okay, so these are the beta coefficients. So we found some negative associations there. We're not totally sure what to make of the CRP, but generally inflammation, bad for growth, as any neonatologist will tell you, right? And also particularly for muscle mass, inflammatory response particularly depresses muscle accretion. And then milk insulin as well. So we have negative associations with fat mass, weight for age, and length for age. Okay, so you can see the scatter here, right? This isn't like a either or on-off switch or something like that. But what we're going to continue to do is look in the aggregate, looking across these things. So there will be women who have particularly high leptin and insulin and CRP maybe, and other factors. And that so we can start looking at kind of how those factors coalesce and how they will be influencing the infant outcomes. So what I think I'm sort of saying up till now might lead you to think that breast milk from obese women is bad. We don't want women to breast. No, that's not what I'm saying. I also have one of my hats is the chair of the Maternal Child Health MPH program in our school. And, you know, people get really hot and bothered when you start questioning breastfeeding or breast milk, right? Because it's, for good reason, promoted as one of the most fundamental, you know, sort of things we can do for health in general is to promote breastfeeding. You know, this is on big time evidence, randomized control trials, right, of breastfeeding support. You get higher IQ, you get lower atopic dermatitis, you get lower gastrointestinal infection rates. It's good, okay? So we're not saying don't breastfeed or that you should be questioning that for obese women. But what we are saying is that we need to know more about what's in milk and how to optimize that milk, hopefully, for the mom through her nutrition, maybe through modifiable factors during pregnancy. Maybe the gestational weight gain limitation could benefit. Maybe the postpartum weight loss, amping that up. Maybe controlling, once we get into diabetics, maybe controlling her insulin, getting metformin going. These are all things that are going to be tools, right, in our toolbox to optimize outcomes. But compared to formula, breast milk is still better. So a secondary research question is going back to that supply issue, because I think that's super important too, and it's probably honestly more actionable for us and might make more of an impact even than the composition side, because we have such a hugely lower prevalence of breastfeeding at all in obese and diabetic women. So I'm going to talk just briefly here at the end about what we're looking at in our study about obesity before pregnancy, glucose during pregnancy, postpartum prolactin, and positioning on milk supply. Getting back to this, I think this is just so fascinating that, okay, these are histological slides from mice. And this is the mammary gland. These are mammary glands during pregnancy and then during lactation. And these are the lean mice, and these are obese mice. And you can see that the structure is really different already during pregnancy, with much more of these adipocytes here. These things here are the ducts. Okay, these are the ducts. So you see this greater development of the ducts than in the lean than obese, clearly a lot more adipocytes. But then when lactation starts, what's dark here, this is that mammary epithelial cell around the alveoli that is in dark here. That's the production floor, right? So much denser than you have here in the obese mouse tissue. There's the lumen where the milk is accumulating. So really different structure. So, you know, and clearly what we know about the production means that doesn't look like it's going to be as effective, all right? And so what we did is we're taking a look at milk output. Getting back to how we do this study, the standardization, we did the standardization so that we get really nice kind of apples to apples comparisons of the milk for every woman. So every woman comes in early in the morning, 8 a.m. It's her second feed of the day. She comes in, we weigh her baby, she feeds her baby ad libitum, we weigh the baby again, and then we wait exactly 2 hours for the milk expression. So we get also this pre-post feeding weight, and that is a classic measure of milk output that's used by lactational counselors to help women and see if they're sort of what their production is, right? Because you can't really tell otherwise without weighing the baby. We just sort of collected that as a side issue. We weren't super focused, right, as I said, on the quantity, but we're taking a look at it. And I think it might have potential as a predictor of cessation. Certainly we see that it's reduced in the ounces that a baby is taking in is significantly lower in the obese moms in our study. And these are all exclusively breastfeeding, healthy moms. Those kids are, during the test feed, had lower intake. We've done it again at 3 months, and we see a high correlation of output, this measure, at both time points. So, when lactation consultants are talking to women about breastfeeding, what they'll say often, and what the sort of accepted wisdom is that low supply is a symptom, not a cause of the lactation problem. Low supply is often secondary to the beginning of supplementation. So when women start supplementing, their demand goes down, so then the supply goes down, right? But, you know, there's also, I think, a factor in which that's bidirectional, so that obesity is hindering lactation and the production of milk as well, which then makes babies maybe be hungry and women get really, really worried when their babies are hungry and crying. And early on in lactation, it's super hard to sort of say, I'm good with the baby coasting and maybe not gaining all that weight right away by 2 weeks. I'm kind of not okay with that, and I'm just going to go ahead and give some formula, right? Because baby's hungry. I'm tired. And so there's a really short period of time after birth when you can sort of be influencing this. And what we're showing here and also here, where the output, this is in grams, is negatively related also to her circulating glucose levels. This is the oral glucose tolerance test results, all non-diabetic women, but negative relationship there. Negative relationship in an in-depth study where we did this at 7 days, same relationship. Lower prolactin at 48 hours in the obese women, and poorer latch. So there's all kinds of stuff happening right here early on before that mom ever goes back for their 2-week visit, where that obese mother may be having some legit physiological issues with lactation that I think could be reducing the supply here. And then that could be promoting supplementation and then further reduction in supply and ultimately cessation of breastfeeding. Okay, so to sort of summarize, we're looking a lot at this. Over here, maternal obesity, her gestational weight gain, maybe some clinical hyperglycemia. We're going to be expanding the study, we hope, in the next phase to be looking at maternal diabetes as well and its control and how that can influence both the milk quality. Here we're talking about CRP, leptin, insulin, adiponectin. We're going to be expanding that to metabolomics, HMOs, as well as milk quality, but also quantity. Really interested in that quantity issue because of its public health significance. And what we think is that what we should be doing, is really, really having much better OB-GYN, easily translatable modules for women that are obese or diabetic, so that some of this education and help and what's to expect right after birth with some of these issues can be in mind right away without waiting for the failure to start and the lactation consultant to have to come in after a week or two, after which points the process has kind of gotten out of control. A little bit of food for thought here, going back to our hormone work. So the ACA provides a breast pump for every mom who wants to breastfeed, and moms are using them. Every single woman in our study, and we didn't say you needed to, every single woman said, oh yeah, I got a breast pump. Oh yeah, I was starting to use it by 6 weeks, if not before. So, and before the ACA, that was beginning to happen, but now it's like totally happening. I mean, pumping breast milk is the way we breastfeed our babies now, for the most part, right? We pump and we store and then we feed later and we freeze and we thaw and we reheat. Donor milk also, incredibly important for our NICU populations. That's frozen, that's thawed, that's rewarmed. What is that doing to all of those proteins in the milk? We have some evidence that actually what we do with the donor milk, which is a Halder pasteurization, it's low temperature, but it's lowish temperature, 62 degrees Celsius. That has some implications for the bioactivity of some of these peptides, right? These hormones. And so something to keep in mind that like the more we understand, the more we can maybe even tweak how we operate with regard to donor milk and with regard to how we help moms properly store and use their own pumped milk. So conclusions. I hope I leave you with the idea that milk is really, really complicated with so many hundreds to thousands of factors in it. And the factors do vary by woman. It's not a uniform substance. Maternal weight status and other aspects of metabolism do appear to influence leptin, insulin, and oligosaccharides, but also perhaps adiponectin and inflammatory factors, and many other features of milk as we're finding in our metabolomic analyses. And those factors, some of those same ones, are associated with altered infant growth and composition. Fewer studies are there. We need larger studies to really understand the aggregate effects of all of those components. But essentially what we are kind of creating is a story about sort of milk as a really important mediator between maternal metabolic health and infant metabolic health. And our ultimate goal is to advance the research so that we can find new ways, sort of new places to intervene to maximize breastfeeding for all women, regardless of BMI or diabetes. I want to make sure to point out that this is a really huge team effort. These are some of the people that work here at the University of Minnesota site. I wanted to point out a shout out to the Center for Neurobehavioral Development. Kristen Sandness has been helping us a lot with the moms and babies. The CNBD is a really happy home for me, intellectually, and also for my lab. And then students, Ghazaleh, Regina, Neely, Kara, they've been amazing in helping get some of these publications out. Jacob is a stalwart data manager who just got his MPH, woohoo! And Lori Foster, I couldn't do this without her. If you ever want an amazing data collection person, think about hiring a family life specialist. She's fantastic. I mean, she gets families and children and how to help them so that what my research subjects get out of the study, yeah, they get a Target gift card, right? But they also get free, repeated patient consultation, because she just does that for everybody, and helping them understand how their baby's growing and what it means and what they can do differently. So that would probably be my other takeaway, is like, hire a family life specialist for your research studies. And then here's my partners in crime, my amazing co-PI, David Fields in Oklahoma, Alicia Coonan-Bateson at Health Partners, and my colleagues here, Lisa Harnack and David Jacobs in Epi, Pat McGovern in the Environmental Health Sciences part of our School of Public Health, and then Tanya Schoenfuss, who is a dairy scientist and has been so helpful in helping me understand things like milk fat and glucose and lactose, et cetera. So that's that.
That was really great, Ellen. Thank you. Did I understand your slides right? Did you say that insulin, CRP, and leptin, that higher levels, like you would find in an obese woman, are actually associated with less body fat in the infants? Mm-hmm.
Yeah.
Does that go against your hypotheses?
Well, so leptin, no. We had a clearer hypothesis on leptin because we know it's appetite suppressing function, right? So we think that the reason why we have high leptin in obese individuals in their serum, right, is because they're not responding to the leptin. So there's that. It's the body's saying, yeah, you've had enough, but you don't stop. So actually, I think that relationship is very expected in infancy, when maybe the regulation of appetite and satiety in the hypothalamus, all those hypothalamic, leptin sensitive neurons are actually still working right, and they're not resistant. CRP, again, I don't know. I think that inflammation, and I think as we look at more inflammatory factors, I think you can make a reasonable case that, well, we know that these factors can cross through the infant gut and into the circulation. But there may be local factors. For CRPs, antimicrobial, actually. A lot of these, immune factors have local effects right on the tissue in the gut. And so maybe there could be something there, that in interaction with the intestinal microbiome or with the tissue of the intestinal epithelium. The insulin, I don't know. Insulin we think of as growth promoting, right? So you'd think that greater insulin, and also we see that in pregnancy, right? So the women with diabetes are going to have higher glucose levels than in the infant, and they're going to be exposed to that, and they're going to grow more, and they're going to be large and fat when they come out. There's also, insulin has a lot of different functions. And so there's some appetite regulating functions for insulin as well. So yeah, I think it's sort of difficult. What do you think, Tony? What are your thoughts? Got it. Right. So you're talking about we're talking about the transmission of obesity. These kids are smaller with higher levels of the leptin and the insulin, which is higher in obese women, right? So what I think is going on, and I wrote a paper with your fellow, Katie Larson-Odie, where we found just an association between maternal obesity and a very transient, early, slower growth in those children as compared to the normal weight women. And I've seen this also published on this in terms of higher birth weight individuals have sort of a postnatal cash down period. And we saw that in Katie's study. And so I think there can be certainly a case to be made. We see this in the Fels longitudinal study too, where over time, birth weight's gone up, but the postpartum, there's been an early, slower sort of down regulation of growth, and then a quicker, earlier adiposity rebound and a greater BMI later. So a sort of change in the shape of the infant growth curve, which is kind of taking it down and then bouncing back up. So again, we need to keep looking at these infants and see what's going on. I'd point to also the Fto gene, the gene product of Fto influences appetite, but even there, those that carry the obesity risk allele in Fto alleles tend to have actually an early infant growth deceleration and then a greater acceleration later. So, in these normal weight individuals who aren't starting super high, like if you think about a kid who's really at birth, they're going to probably be coasting high throughout, right? So, but these are normal weights, and there's all kinds of change in trajectory that are happening in the first 6 months. And so I think that we just need to kind of see if any of these results pan out later. Our prediction is that they're going to go down and then they're going to bounce back higher than the other. Yeah.
Just a question on IL-6 and CRP. Do you have any data, or is there data on how maternal IL-6 or CRP influences those in the infant? So specifically looking at cord blood or some other source that you could get right at birth, or is that just a bad time to measure? No,
that is an okay time to measure, and we do have stories serum from the mother. We didn't get cord blood. Hoping to do that here. It's all of you who do this kind of research, it's cumbersome. Logan had the Riverside birth cohort where he was able to get cord blood, and it's hard. We didn't do that for this large scale study. Would like to, but we do have the maternal serum, and that was sort of part of AIM-3. So we're going to be looking at the correlations there. Is that what you're getting at, between what's in the serum and the cord blood and then what's in the milk? Right. And the reason we do have a subset with that is in this observational study, if those are highly correlated, then the interpretation that it's milk CRP or milk IL-6 or milk leptin, for that matter, that's really driving relationships with the infant could be completely wrong, because it could be the prenatal exposure to those elevated levels that's really driving it. And with an observational study, you have to be thinking about those confounding factors. So we do have the serum, and we're going to be taking a better look at that. There's also the National Children's Study, and they have some stored serum and milk that I hope to get my hands on and look at that too.
So Tony asked my first question, so I'll ask my second. You had an interesting experiment about the mice that were cross-fed based on the leanness of the mice, and alluded to the fact that there's not an equivalent experiment in humans. But that brought to mind that there used to be a thing called wet nursing, which apparently still goes on elsewhere in the world, although it provokes shudders in the U.S. Given any thought to at least observational studies of wet nursing and what the effects of that are?
So I don't know where to find those subjects, honestly, here. It really doesn't happen too much here, but my colleague David Fields, it wasn't going to be wet nursing, but he had a proposal to the Thrasher Foundation, kind of one of those crazy out of the box kind of things. And he said he was going to recruit women who were going to donate milk for this study. And they were all going to be normal weight, non-smoking, healthy. And then he was going to, not randomize, that'd be unethical, but essentially say, okay, obese women, if you want to use donor milk instead, or if you need to, here we've got this donor milk, and he would have, collected it from normal weight women and use that and take the donor milk right over here. Yeah, the Thrasher Foundation said, weird, no, don't like. So they had the shudder feeling about that as well, I guess. But I mean, I think it's honestly, when we know more about what pasteurization might or might not do, the wet nursing might turn out to be a good idea.
Yeah. Did you adjust your models for milk volume?
Yes,
okay. I assume that you did, but it really brings up an interesting point, and was getting my mind racing on this, because I wonder how much programming is going on. So if you start out very early in life not getting enough calories because there's decreased milk production in the mothers with obesity, what kind of programming then is going on? I mean, we see in the children and adolescents that we try to treat with obesity, it's very refractory, right? And they tend to, even with modest amounts of weight loss, their RMR just drops. They are hungrier, less satiated. So, I mean, could this be an important mechanism that if very early on you're doing something to the energy regulatory system that's priming them to want to eat and store calories very efficiently?
Yeah. So you started with, do we include the volume as a covariate? And we do, because we just don't know. And maybe more milk is different. The woman who's producing a lot of milk might have a different composition than those who are producing less. And in fact, I just looked at some data that we have the 1 month milk composition, and then we have incident cessation of exclusive breastfeeding. So because they all were exclusive at 1 month, but there was a small proportion that were not exclusive at 3 months and then at 6 months. And actually there's that milk leptin, milk glucose, milk insulin, and IL-6 are predictive of whether or not she's going to cease breastfeeding. So, and so does output, or, the volume. So some of that volume is all correlated maybe, and that's why we put it in there as a potential confounder. But your other question, which is about programming due to maybe a transient period of insufficient milk and then demand, I don't think that's really been looked at. All right, well, thank you.
You have been disconnected from the meeting.